Pratima from Planet Physiology and welcome to the session on digestion and absorption of foodstuffs. Before going into details of this process, let us study peculiar features of intestine that makes it the predominant site for absorption. As you can note in this picture, inner wall of small intestine has many foldings. These are called as folds of Kerckring or valvulae connivens. These folds are mainly present in duodenum and jejunum, and they increase the surface area of intestinal lumen by 3 times. If you observe the ileum, it will show a much smoother surface as folds of Kerckrings are relatively lesser in proximal ileum and totally absent in distal portion of the ileum. Further, if you see the enlarged endoscopic view of intestine, you will find that the entire lumen of intestine appears velvety in nature, and all these finger-like projections are called as villi. These villi increase the surface area for absorption by 10 folds. Shown in this picture, these are villi. This is the single villus which is cut open to reveal the internal structure. Each villus is lined by a layer of columnar epithelial cells called as enterocytes. Each enterocyte has minute small projections on their apical surface called as microvilli. These microvilli are 1 micron thick and constitute brush border of small intestine. They further increase the surface area available for absorption by 20 times. Thus, there is total increase in the surface area by 600 times, 3 times because of folds of Kerckrings, 10 times due to villi and 20 times because of microvilli. Hence, due to its increased surface area, the major site of absorption of foodstuff is small intestine. Smaller quantity of absorption, mainly water and electrolytes, also occur in colon and even much lesser quantity in stomach. Now let us begin with digestion and absorption of various foodstuffs, and we shall start with carbohydrate digestion. Major source of carbohydrates in our diet is in the form of starches, followed by sugars like sucrose and lactose. Another important source of carbohydrate is cellulose. But human digestive tract does not have enzyme cellulase which digests cellulose, and hence cellulose is excreted in the form of dietary fibers. Let us study how carbohydrates are digested. Carbohydrate digestion begins in oral cavity due to action of salivary amylase. Basically, it is an alpha amylase which acts on starch to convert starch into maltose and small glucose polymers. As the food remains in oral cavity for very short duration, alpha amylase acts only for short duration and is responsible for 5% of total starch digestion. But the small quantity of maltose which is formed in oral cavity gives sweet taste to starch, which is basically tasteless substance. Action of salivary amylase further continues for about an hour or so in stomach until the pH of gastric contents becomes less than 4. During this time, about 30 to 40% of the starch digestion is completed. As chyme passes into small intestine, it gets mixed with pancreatic juice, and now pancreatic amylase acts on remaining starches to convert them into maltose and glucose polymers and causes complete starch digestion within half an hour duration. Most of the starches in the intestine are in the form of maltose or small glucose polymers. Microvilli of the enterocytes possess specific enzymes to convert the partially digested food into its basic components. These enzymes are collectively called as brush border enzymes. These enzymes convert disaccharides to monosaccharides. Lactose is converted into 1 molecule of galactose and 1 molecule of glucose with the help of enzyme lactase. Sucrose is converted to fructose and galactose with the help of enzyme sucrase, and maltose is converted to 2 molecules of glucose with the help of enzyme maltase. As major foodstuff is in the form of starch, it is converted to maltose and thereby into glucose. Thus, the end product of carbohydrate digestion is mainly in the form of glucose, which accounts for about 80% of the total digestive end products of carbohydrates. Remaining 20% are in the form of galactose and fructose. Now let us understand the process of absorption of these end products with the help of a simple line diagram. This rectangle represents enterocyte. This is the lumen of GIT, and therefore this border of the enterocyte is called as luminal border of enterocyte. This side is extracellular fluid, and therefore this border of the enterocyte is called as basal border of enterocyte. Basal border has many sodium-potassium pumps. They pump 3 sodium into the extracellular fluid and 2 potassium into the enterocyte, and therefore they maintain very low concentration of sodium within the enterocyte. This low sodium concentration facilitates entry of sodium from lumen into the enterocyte, and when sodium is entering into the enterocyte, it drags other substances like glucose along with it. Thus, glucose is transported within the enterocyte by the process of cotransport, and the glucose transporter in the luminal border of the enterocyte is SGLT1. Once within the enterocyte, glucose then diffuses out into the extracellular fluid from the basal border by facilitated diffusion via GLUT transporters. Thus, glucose absorption is active process because it is actively absorbed from the luminal surface. Luminal border also possesses another cotransporter called as sodium galactose cotransporter. It moves sodium and galactose molecule into the enterocyte. Galactose then diffuses from basal border into the extracellular fluid by facilitated diffusion. Fructose transport is independent of sodium and it is absorbed from the luminal border by facilitated diffusion. Some molecules of fructose enter the circulation by facilitated diffusion process from the basal border, but some are phosphorylated to form glucose and then are absorbed into the blood by facilitated diffusion in the form of glucose. Fructose transporter at the luminal border of enterocyte is called as GLUT5 transporter. Thus, absorption of glucose and galactose is sodium dependent, but fructose transport is sodium independent. Let us quickly summarize the digestion and absorption process of carbohydrates. Carbohydrate digestion begins in the oral cavity. Salivary amylase begins digestion of starch and it contributes to 5% of total starch digestion. Its action continues in stomach for about 1 hour and causes 30 to 40% of the total starch digestion. Remaining starch digestion is completed in small intestine with the help of pancreatic amylase. Final digestion is done by specific brush border enzymes. Glucose is the main digestive end product and it is absorbed by cotransport. Most of the glucose absorption takes place in jejunum, even though carbohydrates are also absorbed in duodenum as well as ileum. Here we finish with the normal process of carbohydrate digestion and absorption. Let us study what can go wrong with this process. Deficiency of any of brush border enzymes leads to diarrhea as well as bloating and flatulence after sugar ingestion. This is because the product of carbohydrate digestion are mainly disaccharides and these are osmotically active particles. Hence, deficiency of brush border enzymes leads to accumulation of disaccharides into the lumen, which attracts water molecules and volume of intestinal lumen increases, leading to diarrhea. Colonal bacteria act on disaccharides and form various gases, leading to bloating and flatulence after sugar ingestion. Another abnormality of carbohydrate digestion is lactose intolerance. It is caused due to congenital deficiency of enzyme lactase. It is also seen in elderly people. These people with low lactase levels cannot digest milk, and ingestion of milk causes diarrhea and flatulence. For them, yogurt is better option than milk as it contains bacterial lactase which can digest lactose. Another congenital abnormality associated with carbohydrate digestion and absorption is deficiency of SGLT transporter. It affects absorption of glucose and galactose, and again the person suffers from severe diarrhea after carbohydrate intake. Here we finish with the process of carbohydrate digestion and absorption and related clinical aspects. Now let us begin with protein digestion and absorption process. Major sources of proteins in diet are meat, fish, egg, milk, cereals and pulses. Protein digestion begins in stomach. Gastric juice contains enzyme pepsinogen which gets activated to form pepsin in presence of hydrochloric acid. Optimum pH for action of pepsin ranges between 1.6 to 3.2. It causes 10 to 20% of the protein digestion and mainly digests collagen. Protein digestion further continues in small intestine under the influence of pancreatic enzymes. Chyme contains partially digested proteins in the form of proteases, peptones and large polypeptide chains. Endopeptidases like trypsin and chymotrypsin convert these partially digested proteins to smaller polypeptides. They are further converted to dipeptides and tripeptides by carboxypeptidase. Very few proteins are still present in the form of smaller polypeptide chains. As these end products reach microvilli, specific peptidases in brush border further digest them into individual amino acids. These amino acids are transported into the enterocyte by sodium amino acid cotransport. They are absorbed in the circulation from the basal border of enterocyte by the process of diffusion. Enterocyte also absorbs small quantities of dipeptides and tripeptides by active transport. These are then hydrolyzed within the enterocyte by peptidases. Thus, the final end product of protein digestion which are absorbed into the circulation are individual amino acids. Let us study some details about the amino acid cotransporters. Out of all the amino acid cotransporters, 5 cotransporters are sodium dependent, and out of these 3 are sodium amino acid cotransporters, whereas 2 are sodium chloride amino acid cotransporters. There are 2 different varieties of cotransporters which are independent of sodium. Apart from these cotransporters, there are proteins called as pepti-1 transporters. It means peptide transporter variety 1. These transporters are utilized for transport of dipeptides and tripeptides along with hydrogen ions. In case of children, IgA in the milk is absorbed actively by the process of endocytosis. Nucleic acids are also type of proteins, and they are digested by the enzyme nucleases into nucleotides which are further broken down into nucleosides and phosphoric acids at the brush border. These products are then converted to individual sugars, purines and pyrimidines and actively absorbed into enterocyte. Out of total digestive end products of protein, 50% comes from diet, 25% from digestive juices and 25% from desquamated cells. 2 to 5% of the proteins escape the process of digestion and absorption and reach colon. Here they are acted upon by colonic bacteria and are further digested. The main site for protein absorption is duodenum and jejunum, and very small quantities of proteins are also absorbed at the level of ileum. Let us quickly summarize the protein digestion and absorption process. Protein digestion begins in stomach under the influence of pepsin, and it is responsible for 10 to 20% of the protein digestion. Remaining protein digestion is completed in intestine with the help of various pancreatic enzymes like trypsin, chymotrypsin and carboxypeptidase. Final digestion of proteins occurs in brush border of microvilli with the help of specific peptidases. Amino acids are absorbed from entire small intestine, that is duodenum, jejunum and ileum, but pyrimidines are mainly absorbed from upper part of small intestine. Now let us study the clinical aspects related with protein digestion and absorption. Congenital abnormalities of enterokinase are associated with protein malnutrition. Enterokinase is the enzyme necessary for activation of trypsinogen to trypsin, and trypsin is the key enzyme for activation of all proteolytic enzymes. Hence, enterokinase deficiency leads to severe protein malnutrition. We have seen that there are specific transporters for specific types of amino acids. In case of Hartnup disease, there is congenital defect in the transport of neutral amino acids, whereas in case of cystinuria, there is congenital defect in transporters of basic amino acids. So here we finish with protein digestion and absorption process, and let us begin with fat digestion and absorption process. Dietary sources for fat are usually in the form of neutral fats or triglycerides, and these are mainly animal origin. We also ingest fat in the form of cholesterol and cholesterol esters. Fat digestion begins in stomach under the influence of gastric lipase. In case of certain species, there is lingual lipase also, which is secreted in the saliva. Gastric lipase mainly acts on pre-emulsified fats and is responsible for about 30% of the fat digestion. Major site for fat digestion is small intestine. As chyme enters the duodenum, it gets mixed with bile as well as pancreatic juice. Bile salts immediately surround the fat globules and form emulsified fat. As you can note in this picture, this is the fat globule which is surrounded by the bile salts. So this is emulsified fat. These emulsified fats have low surface tension and hence they are easily broken down to smaller fat globules by the gastrointestinal motility. They are still surrounded by bile salts. Thus, emulsification process increases the surface area available for fat digestion. This is essential because enzymes are protein in nature and hence are water soluble, while fats are water insoluble. Due to emulsification, pancreatic lipase can act on the surface of triglycerides and break them into free fatty acids and 2 monoglycerides. Similarly, cholesterol esterase from pancreatic juice converts cholesterol ester into free fatty acid and 2 monoglycerides. These digestive end products, that is free fatty acids and monoglycerides, are also surrounded by bile salts. This structure is now called as mycelium. These are small molecules with 3 to 6 nanometer in diameter and these are negatively charged. Because of smaller size and negative charge, mycelium are made water soluble. Now let us see the process of fat absorption. Due to constant gastrointestinal motility, mycelium gradually move towards the enterocytes. As they come in contact with enterocytes, bile salts are left behind into the lumen and digestive end products of the fat diffuse into the enterocyte. Bile salts again surround new digestive end products to form mycelium and bring them to enterocyte for absorption. This to and fro movement of bile salts between the intestinal lumen and the enterocyte is known as fairing. Absorption of free fatty acids into the circulation depends on their length. For example, short chain fatty acids with less than 10 to 12 carbon atoms are fairly water soluble and hence they are directly absorbed into the portal blood. But long chain fatty acids undergo the process of re-esterification before their absorption. It means free fatty acids combine with monoglycerides to form new molecule of triglycerides within the enterocyte. This process takes place in the smooth endoplasmic reticulum. Then as it moves from rough endoplasmic reticulum and to the Golgi apparatus, protein as well as carbohydrate moieties are added to it. Now the product form is called as chylomicron. Chylomicrons enter the central lacteal of the villi by the process of exocytosis and then enter the circulation via thoracic duct. So here we finish with the process of fat digestion and absorption, and so let us summarize this process quickly. Fat digestion begins in stomach under the influence of gastric lipase. It digests pre-emulsified fat. Major fat digestion takes place in intestine under the influence of pancreatic lipase in presence of bile salts. Fat absorption mainly occurs in duodenum and jejunum. Short chain fatty acids are directly absorbed into the blood, whereas long chain fatty acids undergo re-esterification to form new molecule of triglycerides and then they are absorbed into the blood via lymphatics. Now what can go wrong with this process of fat digestion and absorption? There can be malnutrition of fats and fat soluble vitamins. It can lead to a condition called as steatorrhea, where there is excretion of fatty, bulky, clay colored stool. This is seen in case of pancreatic insufficiency leading to lipase deficiency or in case of biliary obstruction leading to lack of bile salts. Sometimes even the deficiency of bicarbonates or excessive secretion of gastric acid can also lead to steatorrhea. This is because lipase acts optimally when pH is neutral to slightly alkaline. Lack of bicarbonates or hypersecretion of gastric acid cannot maintain optimum pH for the action of lipase and thereby affect fat digestion process. Here we finish with the digestion and absorption of various nutrients. Now let us study the absorption of water and some important minerals and vitamins. We will start with fluid absorption. Usually about 1.5 to 2 liters of the fluid is ingested in the form of food. Various gastrointestinal secretions like salivary secretion, gastric secretion, pancreatic secretion, etc. add 7 liters of fluid to it. Hence, the total quantity of fluid present in gastrointestinal tract in a day is about 9 liters. Out of these 9 liters, 7 to 7.5 liters of the fluid is absorbed in small intestine and 1.5 liters passes to colon. Colon further absorbs water and only about 200 ml of fluid is excreted in feces. Whatever is the fluid load over the GIT, 98% of that fluid is absorbed in various parts of GIT and just 2% is excreted in the form of feces. The next important component that is absorbed from GIT is salt. We ingest about 5 to 8 grams of salt in a day and 20 to 30 grams of the salt are added into it by various gastrointestinal secretions. So total amount of salt in GIT is 20 to 30 grams per day and almost entire amount of salt is absorbed back into the circulation by GIT. Sodium absorption in small intestine is mainly by cotransport process and presence of glucose facilitates its absorption. This knowledge is used in treatment of dehydration. ORS, that is oral rehydration salt, contains salt as well as sugar which favors each other's absorption and also water absorption. This helps to recover the extracellular fluid volume and hence the dehydration. In large intestine, sodium absorption takes place through ENaC, that is epithelial sodium channels. Chloride absorption is passive and mainly occurs in jejunum. In ileum and colon, chlorides are absorbed in exchange for bicarbonate. Ileum as well as colon also possess channels for chloride secretion. These channels are affected by cholera toxin. The toxin enters the enterocyte and increases formation of cyclic AMP, leading to activation of chloride secretion. This in turn stimulates sodium secretion and cause osmosis of water, leading to formation of large quantity of diarrheal fluid. These chloride channels are defective in cystic fibrosis, which is an autosomal disorder. In these patients, intensity of diarrhea is less severe. Now we shall study how bicarbonates are processed in GIT. In upper GI tract, that is duodenum and jejunum, bicarbonates are absorbed, but in ileum and large intestine they are secreted in exchange for chlorides. That is, there is chloride bicarbonate countertransport. This bicarbonate secretion is important for neutralization of acids produced by colonal bacteria. Now coming to the vitamins. As we know, vitamins can be either fat soluble or water soluble. Fat soluble vitamins like vitamin A, D, E and K are absorbed along with fats in upper small intestine. Water soluble vitamins like vitamin B and C are also absorbed from upper part of small intestine, and this absorption is sodium dependent. Only vitamin B12 absorption is sodium independent. It is absorbed in ileum in presence of intrinsic factor. The important minerals we are going to study are calcium and iron. Calcium absorption occurs in duodenum and 30 to 80% of the ingested calcium is absorbed. Amount of calcium that is being absorbed depends on status of calcium requirement. If requirement is more, there is increased synthesis of parathormone and vitamin D and they facilitate calcium absorption. Presence of phosphates or oxalates in the food inhibits calcium absorption. Iron is also absorbed from the duodenum, but the quantity of iron absorbed is just 3 to 6% of the daily iron ingestion. It also depends on the requirement. If the iron requirement is more, absorption is more. Presence of vitamin C keeps iron in ferrous state and facilitates iron absorption because iron is always absorbed in ferrous state, and this is the importance of having a piece of lemon in the diet. Presence of phosphates, oxalates or phytates in food prevents iron absorption. Phytates are mainly present in green leafy vegetables, and to nullify this action of phytates, if we add lemon to the food, iron absorption can be improved. Here we have finished with the process of digestion and absorption of various components in the food. Now let us understand malabsorption syndrome. Failure of digestion and absorption process in small intestine results in malabsorption syndrome. It can occur in various conditions like surgical removal of small intestine, presence of gastrocolic fistula, sprue, celiac disease, enteritis or Crohn's disease. What are the features of malabsorption syndrome? There is decreased absorption of nutrients and vitamins, and as a result, person loses weight. As digestive end products are osmotically active particles, they initiate osmosis leading to fluid overload in GIT. Thus, results into diarrhea. Person also suffers from abdominal pain. That's all for this session. Thank you. If you enjoy my sessions, press the like button and share it with your friends. If you haven't yet subscribed my channel, press the subscribe button. To get notifications about new releases, press bell icon. Thank you for watching and see you in the next video.